Rhynia. Rhynia is a fossil form of ancient pteridophyte found in the Devonian chart in Aberdeenshire area of Scotland, and Rhynia or species along with other type of plants and animals found in this chert bed. These plants, especially fungi that is found along with the Rhynia species, used to devour on these living as well as dead Rhynia. It is believed that the arthropod that is found in the same chart bed also fed on the spores of Rhynia. So these plant Rhynia, only two species, that is Rhynia Gwen vaugani and Rhynia major, has been found along with other type of pteridophyte, ancient pteridophyte such as Yarovia, Cooksonia. Hello friends, I am Dr. Ayantika Das, and in this video I am going to tell you about the overview, morphology and anatomy of a fossil, ancient fossil pteridophyte, that is Rhynia species. Taxonomic position. Rhynia belonged to division Pteridophyta and subdivision Psilophytopsida, class Rhyniopsida, order Psilophytales or Rhyniales, whichever university follows, and belong to Rhyniaceae. Basically, there are two genus of Rhynia found. One is Rhynia major and another is Rhynia Gwen vaugani. So by habit, Rhynia is a terrestrial pteridophyte, which means that it will have a vascular bundle, which is a much advanced character than the bryophytes. So basically it is a pteridophyte, primitive type of pteridophyte and terrestrial form, a very old form, that is because it is found in the Devonian period. In the fossils of in the chart bed that belong to the Devonian period it was found. So we see that it is a much primitive type of pteridophyte, and this is the picture of Rhynia, how the fossil looked like, where we see that they have a horizontal creeping rhizome area that branches by dichotomy, and the aerial branch, this one, it is green and further branches dichotomously to form this this fertile zone and the sterile zone. So this fertile zone is basically a sporangia which contains spores that are haploid, dysbiotic spores. And so this is basically a sporophytic generation, and the horizontally growing part, this one is believed to be rhizomatous in function, and here the little black colored region, this one here, it contains a tuft of rhizoids, which are basically functions of root, as because root is not absent, rhizoid, unicellular or multicellular rhizoids, they help in absorption of the absorption of water and minerals, and also attachment of Rhynia. So this is basically how Rhynia looks, and its habit is, these fossils were found near the volcanic region, and still now this region have a high abundance of hot springs. These this silica rich area, they contained this Rhynia fossils. So basically they were tolerant to a very high amount of silica and also high amount of acidic condition. So this is its habitat. And what is its geographical location where the fossils were found? These fossils were found, they were first discovered by Kiddstone and Lang in Aberdeenshire area of Scotland. And two species found. Two species, one is Rhynia major and another is Gwen vaugani. So this major, the first one, major has been now shifted to Agalophyton major. Why? Due to some reasons that I'll discuss in the upcoming slides. So this Rhynia major is now Agalophyton major, and this Rhynia along with Rhynia, another vascular plant fossil, that is the Asteroxylon, was also found. Apart from this Asteroxylon, Rhynia and Agalophyton, we can also find in fungus, fossil of this fungus, Paleomyces. And it is believed that this Paleomyces fungi had a relation, their food feeding relation with Rhynia. It fed on the living plant of Rhynia as well as the decomposed plant of Rhynia, and also the same chert bed contained arthropod fossil of the same period, and this arthropod was also believed to be feeding on the sporophyte as well as the sporangia of Rhynia. Now, in Rhynia, the entire plant body is divided into two systems, that is the prostate system, where it grows close to the surface of the substratum, grows horizontally. Another is the aerial system, which grows vertically upward. So aerial system is whether it is Rhynia major or Rhynia Gwen vaugani, the aerial system is greenish in color and cylindrical, and it is branched. The aerial system, especially here, this one is Rhynia major, A is Rhynia major and B Rhynia Gwen vaugani. So in both the system we see that there is branching. In Rhynia major it is dichotomous, and in here Gwen vaugani, here in B, we see that the branching is of lateral. And so we see these two type of branching depending upon species. In Gwen vaugani, that is here the branching, it is lateral, and these lateral branches, they do not have vascular connection with the main plant, and these branches are deciduous. They fall and they give rise to another plant. So this is a very common method of propagation, clonal propagation of Rhynia Gwen vaugani. So we see that both species of Rhynia, they have two type of branching. The first one contains that is a Rhynia major, has a dichotomous branching, whereas Rhynia Gwen vaugani, that is the diagram B, has a lateral type of branching. Whatever be the type of branching, the tip of the branches, that is the terminal region, it is tapering, and when it attains maturity, sexual maturity, then it bears a sporangia. These structures, these sporangia can be slightly cylindrical to ovate to various shape, but depending upon the species, the dimension of the sporangia changes. Dimension of the sporangia is basically if this is the sporangia, so the length as well as its breadth, it will depend upon what the species is. So every species, for example, the sporangia of Rhynia major is quite different from that of the dimension of the sporangia of Rhynia when Vaughan. So their dimension of the sporangia is dependent upon species. So is the dimension of this this cylindrical aerial branch. The cylindrical aerial branch, their length as well as their diameter, it will depend upon the species. Rhynia major has a quite different dimension of the aerial branches than that of the Rhynia vaughan. Now, this was all about aerial, but the prostate system, this one, which grows horizontally to the surface, parallel to the surface of the substratum. This area, this rhizome region does not have any root-like structure. They have certain tuft of rhizoids on the ventral or the lower surface, and the no other external protuberance is found in the rhizome except the rhizoids, and the vegetative aerial shoot, they also will usually not contain any other leaf-like appendages.
Rhynia,
But
both the
certain
vertical region, that
small
is
hemispherical
the green
projections
aerial
can
portion,
be
as
found,
well as the horizontal
and this
portion,
was
that
all
is the
about the
rhizome.
morphology.
In transverse section, they look similar. Uh, the stem or rhizome or the vegetative aerial portion, it has got an outer epidermal region. Here we see that I am drawing in red. This portion is the outer epidermis. We see that this portion has got a single layer of epidermis which is covered by this thick black colored, we see this is the cuticle, and this epidermis, single layered epidermis, it is interrupted by stomata, and this stomata, it is present only in the vertical region. So vertical region has got stomata, but the rhizome, that is the horizontal region, this one does not have stomata. So stomata is absent in the horizontal system. But the stomata, it is made up of, if this is the pore of the stomata, it has got two guard cells, and surrounding the two guard cells are the subsidiary cells. The subsidiary cells are many in number. So this is how the single layered epidermis with cuticle layer above and containing stomata in the aerial portion. Next to it is the cortex, and the cortical region in Rhynia species is divided into outer cortex and inner cortex. Now I am drawing the outer cortex in yellow. This region that I am drawing here, sorry, not this region, this region, this entire region, just next to the epidermis, this entire region of closely packed, we see that this entire region does not have any intercellular space. So these parenchymatous outer cortex without any intercellular space and single layer is also called the outer cortex or hypodermis. And next to the hypodermis or the outer cortex is the inner cortex that I am drawing in green. See, from this region to this region, this massive region is the inner cortex. Note that the inner cortical region has parenchymatous cell, but the parenchymatous cell is very massive amount of inner cortex, and this inner cortical cells, the parenchymatous cells, they are greenish. That is, it will have a photosynthetic function. That is, inner cortex is the photosynthetic area, and the outer cortex is just protective in function. So next to the inner cortex, I'm drawing in red now again. Next to the inner cortex is the stellar region, and we do not have any endodermis or pericycle surrounding the stellar region. So this region is the stellar region, where the outer phloem is surrounded by the central xylem. So this dark colored cell is, these are the xylem. So this type of stellar region, it is protostelic with central xylem and phloem surrounding the central xylem. And the xylem is mostly tracheids, and in this type of tracheids we see that both annular as well as spiral type of thickening is found in their cell wall. And the phloem that is present outer to the xylem, they are massive compared to the xylem, and this phloem region, they have a sieve area type of cells. So both phloem and xylem present in the stele of Rhynia, but the stele of Rhynia do not have any
So now
endodermis
about
or
the
pericycle
sporangia.
surrounding.
We see this one is the fossil sporangia, and the picture A shows the sporangia of Rhynia major, and picture B is of Rhynia Gwen vaugani. And these are basically how the spores look. So we see that the sporangia, it is almost slightly tapering and cylindrical, not completely cylindrical, and also the dimension, that is the length, as well as the thickness, that is this one, thickness as well as the length of the sporangia, it will depend upon the species, that is whether it is Rhynia major or Rhynia Gwen vaugani. Upon that, the dimension of the sporangia will look, and the sporangia contains these spores, this tetrad of spores. Actually this here it is only 3 spores are seen, but another spore is present that is hidden from view. So this tetrad of spore, and these spores are haploid, that is N, and this the sporangia, this which contains numerous spore, it is having a stalk and a capsule region, and the capsule wall, this region, the capsule wall is multi-layered. The very outermost epidermis is a single layer, and next to the single layered outer epidermis, which is also very cutie, having a thick layer of cutin outer epidermis, and next to the outer epidermis is the middle parenchymatous layer, and this parenchymatous layer contain inner to the parenchymatous layer is the tapental layer, and this tapental layer is mostly nutritive in function. So if we see that the sporangial wall has an outer epidermal region which is cuticularized, and middle few palisade parenchymatous region, and the innermost is a tapental region. So this is the multi-layered spore wall of the sporangia, and the sporangial cavity, it contains numerous spores, and here we see that in this type of sporangia, the spores are present freely without any structure like columella or elaters. This type of structures are absent, only spores are present, and the spores are generally 60 micron in diameter, and they are homosporous, that is same type of spores are present, and they have a triradiate mark. So this type of marking will be present
in the spores, and these triradiate mark are the areas from where the exine will rupture. So these type of spore, they have a triradiate mark, and the spore wall is also very heavily cutinized. So we see that this type of plant, Rhynia, has a cutinized epidermal region on the plant itself, on the sporangia, as well as the spore. So it is well protected to survive desiccation. The dehiscence mechanism, that is the process by which the sporangia dehisce, it is not known, but it is believed that the sporangial wall, they degrade, they desiccate and they degrade, and only after the disintegration of the outer sporangial wall, the spores are liberated.
Gametophyte of Rhynia. The gametophyte of Rhynia, it looks like a tuft of filaments, and it is very fragile. In fact, this gametophyte of Rhynia bears similarity with other plants like Agalophyton, Horneophyton, and Nothia, where this tuft of filaments, they bear at their tip male and female gametangia, and this gametophyte was first discovered by Kerpetol and later Marker and Panth. In fact, Panth has an opinion that the Rhynia Gwen vaugani, the gametophytic axis, is actually the Rhynia major. And but this type of gametophyte, it is very rare, and also it is much debated, because Spore in 1966, he did not have the same opinion as that of Karp, Merker or Panth, and he said that this Anthridia and Archegonia fossil form were not found in Rhynia. So it is a hypothesis that these are the gametophyte. They look like this, but actually we didn't have that. Fossil form. So this is just an hypothesis. So gametophytic form is just an hypothesis according to Spore. So this is the life cycle, where we see this is the sporophyte, that is the 2N plant, the diploid plant. This is the sporophyte plant, which bears on its terminal branches, on the apex of the terminal branches, it bears the sporangium, and this sporangium inside contains the spore. So basically the spore is haploid, and the spore on germination, actually the spore is dispersed by disintegration of the sporangial wall, and it falls on suitable substratum, and this type of gametophytic plant is produced, where we see that this is the male gametophyte, and this one is the female gametophytic plant. And on here, this region, they will contain the Archegonia, the Anthridia. So this is how the Archegonia and Anthridia look like, and they produce the Anthridia produces sperm, and Archegonia the egg, and later by fertilization again the sporophytic generation A
is
comparative account
continued.
of the two type of Rhynia, that is Rhynia Gwen vaugani, and Rhynia major. We see that they have separate dimension. So the first difference between them is their dimension, where we see that Gwen vaugani is 20 centimeter tall, that is 20 centimeter in height, and the Rhynia major, it is 20 to 50 centimeter in height. So Rhynia major, it is much taller. It can be much taller than the Rhynia Gwen vaugani, and in diameter
the Rhynia major is also have a higher diameter as compared to Gwen vaugani. And the second point is that the difference is that the Rhynia Gwen vaugani, they have a lateral type of branching, and these lateral branches, they do not have any vascular connection with the central axis, so they are deciduous and they are actually used for clonal propagation. They drop off from the plant and new plant is formed from the cast away from the lateral branch that is dropped off, that is deciduous. And in Rhynia major here, this type of lateral branching is not present. Branching is always dichotomous. So this is the second point of difference. First point being the dimension, second point is the branching, and about the third point, Rhynia major. Now Rhynia major has a very different vasculature as compared to the Gwen vaugani, because here the xylem, that is the tracheids that is present in the xylem, it does not have secondary thickening in their cell wall. So as the xylem is very primitive, in fact it looks like the hydroids that is present in the bryophytes and the mosses. Basically hydroids are cells which help in transport of minerals and water, but they do not have a thickened wall. So these type of hydroid type of cells is basically present in Rhynia major, and in Rhynia Gwen vaugani. Here Rhynia Gwen vaugani contains trachet, but Rhynia major, it does not contain trachet in their xylem, but basically hydroid type of cell. And due to this fact, the Rhynia major is now included in a new genus, that is the Agalophyton, and no more it is considered to be Rhynia. So basically Rhynia, now if we consider Rhynia, we will only consider Rhynia Gwen vaugani. And also Rhynia major is also found, apart from Aberdeenshire, it is also found in America. It is also found in the Scandinavian countries like
between
Norway.
these two Rhynia,
And
now both these
it is
type
also
of
found in
two type of
Belgium.
Rhynia,
So
that is
this is
Rhynia
basically
Gwen vaugani
the
and Rhynia
difference
major.
between
They
Rhynia
have
Gwen vaugani
they
and
are both
Rhynia
rootless, that is they
major.
have rhizomes, not roots, and they have a creeping rhizome. Instead of the roots they have the rhizoids, and rhizoids are born on the creeping system called the rhizome, and this creeping system or the rhizome bear aerial system, that is the leafless axis. And these leafless axes in both species of Rhynia, they are photosynthetic, that is they are green, and they branch several times. The tip of these branches they taper, and when they reach maturity they bear the sporangia also. So not only this Rhynia Gwen vaugani and Rhynia major, they have similarity with each other, but also they have similarity with another pteridophyte, that is the Horneophyton, where they look similar to the Horneophyton, but only difference is in the sporangia. The sporangia of Horneophyton is cylindrical, that is like this. It will be like this. But here in Rhynia, we see that the sporangia is much tapering, like this type of sporangia. This type of sporangia is present in Rhynia, and this type of sporangia is present in Horneophyton. So only difference is the sporangia. Other part of the plant looks similar. Also Rhynia bear similarity with asteroxylon, but only thing is that they have a kidney shaped sporangia, and that kidney shaped sporangia is borne along the stem. This type of sporangia is present to be borne along the entire stem, not only limited to the apical region.
And also now Rhynia major is renamed as Agalophyton, and it's a new species. So this we say that Rhynia when vaugani is also similar to the Agalophyton. So that was all about Rhynia species. My next video will be on Botriopteris, which Vaishali Mishra has requested me. If you want me to make any video on any topic, do write it in the comment section and I will be making those videos one by one. And if you like this video, please do share it with your friends and do subscribe because your subscription really encourages me. And those who have already subscribed, thanks for subscription. And I have made videos on algae, fungi, on bryophyte, pteridophyte, on lichens. So you can go to my channel and watch those videos. All the pteridophyte videos link I have put in the description box. So thanks again for watching my videos.